Welcome to my channel. I'll be making another video in which I'll be discussing some MCQs related to microbiology examinations for lab technologists and for other examinations for DHA, MOH, and Haad. So, the first question is septic collection of blood cultures requires that the skin be cleaned with 22% iodine, then 70% alcohol solution, 70% alcohol, then 2% iodine. 70% alcohol, then 95% alcohol. D 95% alcohol only. So the answer is A, because 70% alcohol works better than anything else. So first, this realization is done using 70% alcohol, then 2% iodine. When cleansing the skin with alcohol, then iodine for collection of blood culture, the iodine or iodophore should remain intact on the skin for at least 60 seconds. So the questioner is asking the question: when cleansing the skin with alcohol and then iodine for the collection of blood culture, the iodine should be intact on the skin for at least 60 seconds so that no microbes could get in or penetrate inside your skin. What is the purpose of adding 0.025% to 0.050 sodium polynithosulfonate to nutrient broth media for the collection of blood cultures? The answer is A, because it inhibits phagocytosis and complement. So the question again is, what is the purpose of adding SPS to a nutrient broth media for the collection of blood culture? It inhibits phagocytosis and complement. Here is the explanation. SPS is used in commercial blood culture products because it functions as anticoagulant and prevents phagocytosis complement activation. In addition, SPS neutralizes aminoglycoside antibiotics. Addition of SPS may inhibit some Neisseria and Streptococcus, but this can be reversed with 1.2% gelatin. So, SPS as added in blood culture to avoid phagocytosis, and it is an anticoagulant. Fourth question: a flexible calcium alginate naso nasof uh, a nasophyll swab in the collection device of choice for recovery of which organism for the nasopharynx? So, a flexible calcium alginate nasopharyngeal swab is the collection device for which choice of recovery for which organism for the nasopharynx. So the answer is A, Staphylococcus aureus, because when the nasal swab is taken and the device is for the recovery of the organism called S. aureus or Staphylococcus aureus. The answer is a specimen for the culture of N. gonorrhea best if plated immediately or transported in a medium containing activated charcoal to absorb inhibitory substance that hinder the recovery. So the nasal swab is taken for the recovery of Staphylococcus aureus. And the fifth question: semi-solid transport media such as amide, strod, or carrier are suitable for the transport of swabs for culture of host pathogens, except A. Neisseria gonorrhea. Semi-solid transport mediums such as amide, strod, or carrier are suitable for the transport of swabs Acha specimens for the culture of gonorrhea are best if plated immediately, transported in medium containing activated charcoal to absorb inhibitory substance that hinder the recovery. 6. Select the method of choice for recovery of anaerobic bacteria for a deep abscess. Answer is C. Needle aspirate after surface decontamination. Select the method of choice for recovery of anaerobic bacteria for a deep abscess. Answer is needle aspirate after surface decontamination. And explanation: the anaerobic specimens are easily contaminated with organisms present on the skin and mucosal surfaces. When a swab is used, needle aspiration of an abscess following surface decontamination provides the least exposure to ambient oxygen.